myself and Ian Davidson from Cawthron, and Melissa Welch, who I think is also watching the webinar, from an economic modeler from Scion Research. So what I'll be talking about today is really representing our ideas and our progress and our aspirations. Yeah. Cool. So New Zealand's maritime transport network, as an island nation, heavily dependent on imports and exports, New Zealand's coastal environments are a busy place. There's a lot of vessels around our coast at any given time, including thousands of recreational yachts and launches, commercial ships, including passenger vessels, cargo ships, container ships, tankers, ferries, and tourism boats. And also we have a vibrant aquaculture industry, and the operations of which include, movements of equipment and stock and vessels that provide particular services. And all of that together results in a really complex domestic network, maritime network, and for good reason, because economically speaking, it provides the pillars of New Zealand's economy. But also exploring our coasts recreationally obviously makes us appreciate what we've got, and is a lot of fun for a lot of New Zealanders. But also the voyages of different vessel types provide multiple mechanisms for the transport of marine pests and potentially diseases that are already established in New Zealand or that might be entering here in the future. There is domestic ballast water being moved around. There are organisms growing on the hulls or internal pipework of all vessel types. And essentially, vessel movements can facilitate pretty fast transfers of species across large distances and across natural barriers. So pathway management is important for biosecurity operations in New Zealand. But it really requires us understanding the dynamics of this domestic transport network. And that's easier said than done. If you look at this screenshot here on the left, you see the maritime traffic around New Zealand over a 4-month period from January to April this year. Every one of these white or red lines represents the track of a vessel, and you can see there are hundreds or thousands of vessels coming in from overseas, leaving New Zealand to overseas and moving around our coasts. And the movements are so many of them that you can't really actually see a pattern when you get closer to the coast. The inset to the right here is just a zoom into Great Barrier Island, one of the iconic places up in the Hauraki Gulf. You can see also here just thousands of movements of boats and vessels in and out and around the island. And of course, Great Barrier Island is the place of the recent Caulerpa brachypus incursion. So in summary, really, the system is far too complex for us to understand it without the use of quantitative approaches, which brings us to networks and network analysis. So networks are defined as collections of objects, of which some are linked, and graphs are mathematical models of network structures. Specifically, graphs are sets of nodes and edges, where nodes represent the entities of concern and edges the connections or the relationships between these entities. And network analysis really examines functional relationships among nodes in graphs. The pioneer of graph theory was a Swiss mathematician, Leonhard Euler. He lived in a city called Königsberg, which is today's Kaliningrad in Russia. And the city essentially had several parts to it that were divided by the two branches of a river whose name I forgot, but essentially created 7 different parts to the city. And in the 18th century, Euler developed a mathematical methodology to prove that it was impossible to design a walk through Königsberg that involved crossing each of the 7 bridges, but crossing each bridge only once. And to do that, he actually developed a novel method, which became known as graph theory. And today, graph theory and network analysis are major scientific disciplines. So they have a network analysis and theory has a wide range of applications across society and technology. For example, social network analysis is being used to study connections between people and groups and organizations, and it's and to study the flow of information or rumors or political activities through that network. Network science has also got a huge role in medical science and biotechnology, for example, for understanding regulatory and control functions of gene networks or of our nervous system and neural network. In ecology, network science is being used to study ecological food webs, connectivity between biological populations, and also the design and the effectiveness of protected areas and protected area management. And of course, networks connected with epidemiological models are important in guiding and enabling the design of interventions with disease pandemics, including the current COVID-19 pandemic. So what we're doing in our program is we're trying to look at biosecurity in New Zealand through a network lens. In our graph of New Zealand, the nodes are essentially the ports and the marinas and the mooring areas and the natural anchorages along our coasts and islands, as well as aquaculture farms and other operations, marine protected areas, rohe areas of significance to Maori, and other types of places of interest. And we have, in our developing model, hundreds and hundreds of these nodes all around the country. Each node has a number of key attributes. So, for example, which jurisdiction it's located in, its biosecurity status, whether particular pests of interest might be present there or not, its environmental setting, types of habitats and size of these habitats, temperature and salinity, special significance to Maori living in the area, or commercial or ecological importance, of course, if it's an aquaculture farm or a marine protected area. Hydrodynamics and other attributes. The edges in our network that connect the various nodes are the routes and the voyages of commercial and recreational vessel movements that move around the nodes. And our edges also have attributes. So they include, for example, the potential for transport of marine pests in ballast water or as biofouling, the duration or the distance between two nodes or any two nodes and the duration it might take a certain vessel type to cover that distance, the mortality of organisms on or in that vessel in this situation, environmental barriers such as the difference between, in temperature or salinity or other parameters between the origin and the destination site, seasonal patterns, and many more. So essentially, our network consists of nodes and edges that have different attributes that are relevant to organisms and to biosecurity management. Structural network analysis is something we undertook first when we developed our network models. So these structural network analysis can help us understand the roles and relative importance of nodes. And this is very important for biosecurity management. For example, there are different varieties of so-called centrality metrics in networks, and they can tell us about the measure of incursion or downstream spreading potential of any node in the network, or of its importance as a stepping stone that connect to other sites that are themselves not connected to each other. Some nodes have a higher or a lower degree of influence on the spread of the marine pests across the entire network. And we can also measure things like the ego neighborhood metric, which is and gives us an idea of the reach that a node has through the network via multiple steps. So all these things together really tell us about the relative roles of the different nodes in the network with regard to transmission of invasive species. Even more powerful is the use of dynamic network analysis. And here We can add biological and infection parameters and epidemiological models to the network that we have, and simulate the dispersion of pest species through the network. That can help us predict spread in space and time. And for example, let us know about potential future or present hotspots and hubs of invasion. The dynamic network analysis can also help us design optimized monitoring programs utilizing key monitoring sites. And the analysis of network compartmentalization can help us understand the importance of regional, interregional, or national management approaches of the overall network. So that was kind of the context to what we do. Our team has several objectives. The first one is to develop a domestic maritime transport network model for New Zealand based on 3 pathways: commercial shipping, recreational boating, and the aquaculture industry, and to then apply this model to particular scenarios, incursion and response scenarios, to optimize approaches for prevention and response in the future. This latter objective we do in close collaboration with our end user partners, in particular our 3 regional councils, Northland, Auckland, and Marlborough district councils, and Biosecurity New Zealand MPI, as well as our iwi partners in the program, Patuhara Keke Te Iwi Trust from Northland. So the idea here is to develop scenarios that we're concerned about, either of current distributions of marine pests or of anticipated distributions or of unwanted situations that might be arising in the future, model the dispersal of these species through the network, and then evaluate the effectiveness of a range of different prevention or control or intervention options, I guess, cost-benefit optimized on spread and spreading rate and impacts, together with cost-benefit analysis. So over the last 2 years, we have almost finalized the development of a commercial vessel network. The current spatial domain is the mainland of New Zealand and all the surrounding islands, including the subantarctic islands and Kermadec. It comprises merchant vessels, passenger vessels, including ferries and tourism vessels and water taxis, and all sorts of fishing vessels as well. We have voyages that span a 4-year period, altogether more than 220,000 voyages around the coast of New Zealand. They occurred pre-COVID in our data set. We have almost finalized the structural network analysis, like I described it earlier on. So we have now a better understanding already of the different nodes in the network and their relative contributions to biosecurity risk in different ways. And we're currently building dynamic models that incorporate particular species and their life history attributes and how they interact with different vessel types, hull fouling and ballast water and aquaculture operations. We've been really fortunate to work with Auckland Council and MPI and assist a little bit with the provision of useful information towards the Great Barrier Island management of the Calopa brachypus invasion. You can see on the right-hand side there, we've been through Cal Faubel in particular able to develop some hotspot maps based on vessel arrivals to areas. And we have been able to trace the origins and further destinations of hundreds of vessels that visited the areas around Great Barrier Island where Calopa has been recorded, which is hopefully being, going to be used to be able to inform surveillance and response as needed. We also made a lot of progress with developing our recreational vessel layer of the network. We just finished a nationwide survey of all of New Zealand's keeled recreational vessel owners. This was done in collaboration with the New Zealand Marine Operators Association and Yachting New Zealand, as well as nationwide harbor masters and council authorities. We captured more than 25,000 berths and moorings around the country. Almost 2,000 people responded to our online survey and logged over 12,000 destinations that they visited in their vessel over the previous 2 years. So a network or data grooming and quality assurance is currently in process. This is all just a few weeks ago that the survey finished. A network is in development led by Kyle Hilliam, our PhD student. And we're also really stoked because our collaboration with Yacht New Zealand and ZMOA meant that our data can also provide some support for the recreational boating sector by providing information of, where people from different places tend to go most often, how long they stay there. And it also helps agencies like DOC understand vessel visitation rates to the marine protected areas and other things. So we're pretty stoked about this data set. And you can see in the graphs here on the right and on the left, these are the locations that vessel owners logged using an interactive map. And there's over 12,000 of them. So currently we're just trying to get some structure into that. Yeah, over the next 2.5 years, we've got a lot of work. We going to first finalize our developing networks, in particular the aquaculture one, which is also in development, but not quite at the stage of the other ones. And then we're going to work actively with our end user partners from local and central government and other agencies interested in marine biosecurity management to identify model scenarios for target species or target pests, their distributions, and the types of interventions we might want to simulate, some of which are based on other tools developed in this research program, and also over different time horizons. And together with our bioeconomic modelers, we'll then identify cost-benefit optimized strategies for marine pest or risk prevention and response going into the future. And the overall idea, of course, is that this will support resourcing and also prioritization of resources for marine biosecurity management in New Zealand. Our team is actually getting together due to the pandemic for the first time in 2 years in person. So the 7 of us are going to meet up in the North Island from beginning of next week till the end of next week. And we going to progress our work, make plans and strategies for the next 2 years. So we're really looking forward to that. And that's it about pathway management and network modeling, what we're doing and why and where we're at the moment. We have a website that you can, there's the link to that. You can look up, we try and post progress now and then. You can get in touch with people you would like to talk to about any of the work. And yeah, we hope you're interested in this topic, and we'd love to talk and collaborate and interact with you, because the tools we're developing are really meant for the benefit of all New Zealanders. The more input we get and the more feedback we get, the more informed and the more relevant our approach and outputs will be. So thank you very much from me, and over to Xavier.
Thank you, Oli. That was a great talk.
Thanks for saying that.
Very good. Kia ora, everyone. Tēnā koutou katoa, ko Xavier tōku ingoa. I'm really pleased to be here. I'm Xavier. I'm from the Swiss Alps, actually, but ironically, I became a marine biologist as well, such as this guy here, and with a specialty though in molecular phylogenetics, which I used to study coral reefs ecosystem from around the world for about 20+ years. Which brought me to some very nice areas, such as there in Hawaii with my family, where we spent 5 years during my postdoc. Then I took a job at the Cawthron Institute 10 years ago, which coincided with the arrival of high-throughput DNA sequencing technology, and pretty much the beginning of the environmental DNA or eDNA research field, which now, as you may know, is booming everywhere. I am currently involved in 5 MBIE Endeavor programs, and one of which is the Marine Biosecurity Toolbox, in which I sort of coordinate the citizen science and outreach activities. So today I'm really pleased to be able to talk about citizen science a little bit and share what we're doing. So citizen science is a concept that's usually very, very appealing to most people. For example, for researchers such as myself, I really love the idea that I can reach out to the local community that can help me gather more scientific data cheaply, and that can also help me disseminate these data more widely to my somehow restricted world of scientific publication or scientific conference sometimes. And so this knowledge can get a little bit broader, increase the general knowledge, and then I get to learn many new things on my own project by looking at my project under a very different lens. And it's a wonderful concept, really. But as many of you, if you've been involved in citizen science, you will know that there are these 4 challenges that are not to be underestimated. It requires good logistics. You need to be organized. Engagement requires time, and time equals money. And that engagement really needs to be an ongoing relationship. You need to have this ongoing communication between the scientist and the citizen, otherwise the program just dies off. And then, of course, there is these risks of data biases. So you need to come up with really solid protocols to ensure delivery. So what are we doing in this space as part of the program? So as Ollie said, there's 4 components, but one of them is Detect here, which is all about improving the use of eDNA technologies to empower government, Tangata Whenua, industry, and the general public in a more effective biosecurity, to mitigate more effectively biosecurity risks. And citizen science, the citizen science component of this program really aims to bring easy to use field deployable instruments right in the hands of everyday people. And it's a really exciting period of time right now, because these tools are becoming cheaper, faster, more easy to use. And so really we have this grand vision that not in a not too distant future, a lot more New Zealanders will be able to help us use these tools while they go at the beach with the family and rapidly detect marine pests that could help us, scientists and the governments, to more effectively remediate these incursions. So what have we been doing? So one of the first things we've done was to run this focus group back in 2020, and that was done thanks to wonderful Melissa Welch and Richard Yao from Cyan, and I've seen Melissa is here, so hi Melissa, who put this really nice focus group together in collaboration with 2 Nelson colleges, the Nelson College and Nelson College for Girls. So the idea there was to bring 11 participants into this classroom there, a number of adults and students, and throw at them 9 focus group questions, multiple choice, that were all about gathering understanding what would be the main factors for citizens to actually engage into citizen science, but in the space of marine biosecurity context. And so those questions ranged about, questions around what would be the main drivers for them to engage. Other questions were around what would be their preferred approaches or platforms to use. The preferred season, the frequency, and what sort of incentives do they need to participate. And just as an example here, you see one of the questions. It was quite interesting here to see that when you ask them what would be the main drivers for them to participate, you can see that knowledge, increasing knowledge, education, and rewards, which is not to be underestimated. So reward either in the term of paying them or giving them vouchers or giving them points towards an achievement of some sort, predominated over technology and eDNA in that particular question. But interestingly, on another question about the preferred approach, then we could see this, this shift between adults and students. The student would prefer technologies and eDNA, whereas the adults preferred opportunistic traditional method. So that was a very short and small survey, but very important for us as we're gathering now our thoughts to extend this survey into a nationwide survey. We would really like to push this early next year to get a better sense about broader New Zealanders. Next were the point of need tools. So these point of need tools are simplified eDNA tools that can be used easily and rapidly. This particular tool here is called RPA. It's recombinase polymerase amplification technology, which was put together validated by this person, Martin Zierengimbel. He was part of our team. He worked with us for over a year. It was amazing. And he essentially developed this test, which is a rapid test for detecting the presence of Sabella spallanzanii from environmental DNA samples. It's a very similar concept to the rat COVID test that you're all very familiar with. But this one uses a slightly different technology, which is a PCR that can actually work at body temperature around 37 Celsius degrees. So what you do is you add a little bit of eDNA with or without the presence of Sabella into a tube, you mix it with a bunch of reagents, you hold it in your hands, or even better, under your armpits for 20 minutes. And then if Sabella is detected, and then you transfer, a little bit of liquid onto a lateral flow strip, and then you look at the result, just like the rat COVID test. So Martin did not only validated that tool, but also tested a simplified protocols with 3 distinct group of people with no scientific backgrounds. And what they had to do was they were given 4 blank samples with or without Sabella. They didn't know which one. And then they had to follow this protocol and make the test work without any supports from the scientists. And that went really, really well. The results were fascinating. We learned a lot and how to now simplify the technique a bit more. And it's just about to be published in Frontiers. Another exciting package that we've explored was to organize and embark on 2 research cruises last year. One in Fiordlands and one in Northlands. It was made possible by a really wonderful group of people, and you can see some of the logo or the organization involved here. But perhaps more interestingly is that we think it's one, if not the only time that 2 significant MBIE Endeavour programs actually got together to work together on doing research on such expeditions. So on the one hand, the Marine Biosecurity Toolbox, and on the other hand, the AIM2 program, the Aotearoa Immigration, that's a good one, Aotearoa Impact and Mitigation of Microplastics, that got together. And during this cruise, we essentially collected eDNA and microplastics and actively engage also with a number of stakeholders and users, the general public, to shed the light on the research. So for example, on the boats, what we were doing on the one hand, the biosecurity team was very busy collecting, testing different collection methods or isolation method to isolate eDNA for biosecurity purposes, but also very excitedly, we started using these cutting edge tools right there on the boat. So we were able to extract the DNA, RNA, etc., to run qPCR on a range of pests, to also sequence here high throughput sequencing right there on the boats using this Oxford nanopore sequencing technology. And on the while we were doing this, our colleagues there from the AIM2 team were really collecting a lot of microplastics particles from around the coastlines. And that's a big endeavor to do. It's a lot of work and very important because we know very little on the extent of microplastics pollution in New Zealand. And that's one of the main aim of AIM 2. Once on land, we reached out to various communities. For example, here at the Marine Discovery Center in Lee, we welcomed about 40 local school kids and their teacher who were immersed into the world of microplastics. So we were very lucky to have these wonderful people here from Algalita South Pacific, the Blue Cradle Foundation, and ESR, who put together an incredible show to and activities here that were exposing the kids to understanding what are the different types of plastics in the environment, you know, their buoyancy in the aquatic environments, and their impact on our natural environment. And this was absolutely amazing, very successful. It worked really well with this age group, 7 to 11 years old, these kids, and successfully raised the awareness, their awareness on the urgent needs for ocean protection in that space. And these activities are really great because they can be used now on very different situations with different publics, even adults. It's amazing. We also ran a fantastic workshop here in Whangarei with participants from Northland Regional Council, MPI, DOC, Science, universities, and local iwi hapu. And the particularity of this group is that many of them are actually actively involved in citizen science activities. That group on that day had the opportunity to have some hands-on experience with eDNA tools, but most importantly, we discussed how to best move forward in the citizen science space here in New Zealand, and to better understand how many groups are they, what people are doing, and start to strategize over consolidating a funding strategy. It also made us realize that there's lots of citizen science activities happening, and some of these activities are replicating each other a little bit, and they don't necessarily know of each other. And somehow more frustratingly is that sometimes, and most of times, in fact, those citizen science programs are funded through a short period of time. For example, the Marine Biosecurity Toolbox program is 5 years. So how do we actually establish and maintain such programs? So for us, it's really important to focus, use wisely that money that we were allocated to make a difference. So for us, our preferred approach has been to place most of our energy into schools. And we decided to start small and involve teachers first. And I'd like to thank here Natalie Trigedga and Johnny Fraser from both Nelson College for Girls and Nelson College, who really embarked on this adventure and were really helpful with us. So what we've done is bring the teachers together. They were exposed to a range of activities that we had elaborated, and it was really great because it allowed them to figure out how these activities could possibly fit into the curriculum of their schools. So they got to look at biofouling under the microscope as well, extract DNA, run qPCR, or even play with this underwater drone in a marina that goes and look at biofouling material, for example. And for us, what was really important is that it allowed us to better understand how schools work, because we have no idea. As a scientist, I had no idea how complex the school system works here in New Zealand. So that was really important. And then we started to try out these activities, and they are both indoors and outdoor. So for just a couple of examples, there's many more, but a couple of examples of indoor activities. On the one hand, one that worked really, really well was to go here to the left on Nelson College for Girls, where we have some tailored presentation for these kids to introduce marine biosecurity concept into the classroom, but also bring biofouling material right there. That's works really, really well. It really attracts attention. They learn about the ecology and the marine pests and all that. This is really great. And the other cool indoor activity that we've done to the right here was at the Nelson College. We ran an actual laboratory, like bringing cutting edge tools right there in the classroom. We did 3 sessions. The first session, the kids themselves had the opportunity to extract nucleic acids, both DNA and RNA, right there in the classroom from a range of archived marine pests that we brought there, using this little instrument there. It's the size of a toaster. It's amazing. It's a 1-step, 20 minutes, and it works wonderfully well. I mean, it's incredible. Then on session 2, they use those extracted nucleic acids, and they actually ran their own qPCR on a range of marine pests right there in the classroom. And on the 3rd session, they actually got to see and experience high throughput sequencing right there in the classroom. We believe it's a New Zealand first. We've never heard of that before, so that was quite exciting for us to do. And for me, one of the highlights has been that one of the school kids actually made a little movie. So he filmed and interviewed his schoolmates, the teachers, the scientists, and created this little video. I encourage you to have a look at it. It's quite cool. It's on our news section of the Biosecurity Toolbox website. Outdoor activities, there have been many, but just as an example, here, I don't know if you've heard of this exciting aquabot situation that's going on in the region in particular. So there is the Ministry of Inspiration, led by Amy Cornelissen, is all about these aquabots. So it's these little bots that you can actually build in different ways, and then you can put them in aquatic environments, and they can go up and down, take objects and stuff like that. But here, we thought that we would push this a little bit further, and we collaborated with the Wilder Lab in Wellington, who provided us with these little plankton nets that we placed at the back of this little aquabot. This is in Mistletoe Bay. There were about 20 kids from different colleges around the top of the South, and they actually collected eDNA right there while playing with the bots. And then the Wilder Lab actually sequenced those data and produced this wonderful wheel of life that you can see. It's really powerful. You can see actually all of the organisms of taxa that were actually collected in these samples. And so, so there's going to be a follow-up workshop with these same kids in June here, where we'll show them the results, the sequence data, and how to analyze them. So it's quite exciting in that space. So just to wrap up, I gotta say that now we're pretty pleased because we do have a variety of indoor and outdoor scientific activities, but organized around these 3 sort of components here. But what we really would like to see is to see them gradually incorporated into the school curriculum and maintained and possibly extended in the longer term. So recently, we've been really very lucky to welcome Geert Benke at the Cochrane Institute, thanks to a recent prestigious award from the Royal Society Teaching Leadership Program. So my understanding is that there's only about 6 teachers nationwide that get these grants. And so that allows her to come at the Cochrane and work with us to help us in that space directly. Geert is a biology and science teacher at Nayland College, and she's been there for 17 years. So that means that not only she's very well aware of the curriculum of the school system itself, but she's also very well connected and respected with across other colleges and with many other teachers. And she's been, since she's been here, she's been already doing an amazing amount of work by reaching out to 3 different colleges. And she actually is now putting together specific biosecurity units within the classrooms here, but which will contain some of these activities that we have defined, but also add additional components. And I love this because the idea here is not just do eDNA or biosecurity or observation of biofighting, but put biosecurity context into art, into writing, into visual observation, into communication and blogs, things like that. So it would be a very kind of, a variety of activities that would come together. And the idea is to provide the schools with a package of activities that actually teacher years after years can use without us scientists having to go all the time into the classroom. And also, and I see we're just 10 minutes away, so I'll wrap up really fast, but she also put a pre and post knowledge survey to assess the knowledge of the kids before and after the unit, which is awesome. And very importantly, she's pushing strongly for incorporating, merging Western and Maori science together, which is very important. It's part of the new NCEA school standards that are coming up in 24. A strong push towards integration of Matauranga Maori into the classroom and Maramatanga, which is learning from different knowledge baskets and communicate it at the end of the unit to the general public through an exam. We would love to have a forum where at the end of the unit where kids have to present their finding to the general public. That's one of the dreams. All right, so yeah, keep watching this space. Lots gonna be happening this year and the next. And if you have any question and anybody is interested to collaborate, please reach out. I'd like to very briefly acknowledge my partners in crime that have been very amazing to work with and that help a lot in these outreach activities. So Anastasia, Ula, Johan, Michelle, Geertz, and Martin. And with that, I'd like to thank you for your attention, and we'll be happy to take any question now.
Thank you, Xavier, and thank you, Ollie. Any questions? Just stick your hand up or unmute, and away you go. Just while we're waiting for that, I had a couple myself, guys. Thank you. Great talks, very different sorts of things. So Ollie, first, I was just fascinated when you did your network, looking at the structural network analysis, that Picton was coming up as a more significant node in terms of connection than Nelson, which is not the way we've tended to think about it. What do you make of that?
Yeah, good observation, Peter. I think it's something we noticed as well, and I think it's to do with the fact that the Wellington ferry goes in and out of there multiple thousand times a year. So that's something we might look at excluding or just seeing how that changes its influence, but it's to do with being a domestic, connected to the North Island, essentially.
Right, thank you. Anybody else? I'll keep going just a minute. Xavier, just I was really interested in your survey, getting to understand what people respond to, which we guess about quite a lot, but you're actually getting data about that. Do you see that might be useful in terms of social marketing? Because a key thing for us here is being able to change people's behavior. Are you going to be able to help us with that?
Yeah, absolutely. I mean, that's the grand vision of having this nationwide survey extended. So that first survey was actually, so you can see more of the results of this survey if you go on the website, on the news section. There was a little news allocated to this, so you can see more of the what people preferred or not, the tendencies, the trends, if you want. But clearly, we need to extend that to a much wider range of people and cultural backgrounds nationwide. And yes, once this is done, then I mean we're hoping to even publish this, to put that into a publications, and that would help us to really kind of guide the energy and the direction that we're taking with these activities, because it will dictate what tool you're actually pushing forward and what end users you're actually connecting to make this happen. Yeah.
Great, thank you. And I've got questions for Natalia. You can ask those now, Natalia, if you just unmute.
Hi, that was a really, really awesome presentation, both of them. So with the first presentation, Ollie, I was looking at the nodes as well and seeing, am I correct in thinking that Opotiki was one of the nodes that was in there, a smaller one just off to the side?
Which one was that?
The nodes, there was a large, like you've got the large impact of what it looked like to me, the Tauranga Harbour and the Bay of Plenty. And then there was one more off to the side. And so I was wondering if that was in the Opotiki area because of the aquaculture going on there?
We don't know, just because we haven't incorporated the aquaculture industry yet into that model. Essentially, the figure I showed there is unfortunately a bit of an earlier draft. It includes the port calls of commercial vessels that were received with a whole lot of AIS data that we procured, and essentially it's to the resolution of the port call data, and that's purely commercial vessels.
Okay.
And not aquaculture operations. So, but we can't wait to have the aquaculture layer and model available too, and then I guess superimpose the three together and see what the whole picture looks like and what the relative roles are of each of them.
And will the information be will different industries have to seek that information in terms of, will they need to contact yourself and your team directly, or how will the information feed out?
I guess, it's public funded science, so our outputs are available. We're going to reach out for like this, and via science publications and conferences. We'll probably go to different organizations and provide updates, and say to the aquaculture to the aquaculture industry, to the recreational vessel industry and commercial shipping. And I don't see why we wouldn't be able to provide data summaries. There's some information, that we have confidentiality clauses around a lot of the recreational vessel data. So that's all anonymous, but general patterns and stuff should be information that we will be trying to share as widely as we can.
Awesome. I should have introduced myself. I don't think I can have, I think I can have a video. There we are. Ah. So I'm working from home today, but I work for the Department of Conservation, and I'm I like keeping up with everything. I'm a community ranger, so my role in the Bay of Plenty is to just have an overview of what's going on everywhere. And because I'm in the Tauranga district, the marine area is, of course, quite a big one. And we work with an organisation called Tauranga Moana Biosecurity Capital. So it's quite interesting to see what else is happening around the world. And Tauranga, New Zealand world. So that's cool. So it's kind of, knowing what's coming forward and what's coming ahead. And then, I'm worried I'm going to say your name. Oh, no, I was thinking of your last name. So Xavier, it was quite exciting, your citizen science that you're doing as well with the schools. We have citizen science over here. I guess what's offered is like litter intelligence, those kinds of programs. But I'm interested with how you got into the schools. How did you select the children?
That was a long time making, but because over the years we've kind of had a number of relationships with local teachers. So, for example, Johnny Fraser, which I mentioned there, he also was awarded a few years ago. He was awarded one of these grants from the Royal Society, this exchange. So he came at Coulter, and that's where we met. And then we always had this ongoing communication about how do we bring science a bit more into the classroom, like, linking these two worlds a bit better. And so that's essentially where it started. And then the program came in as part of the Marine Biosecurity Toolbox program. We decided to make a big push there. And that's when we really then connected locally. And when I said we start small, it was important because you can very rapidly lose yourself. If you start to reach out around the nation, different schools, you open the Pandora box right rapidly.
Yes.
So we decided, like, let's be clever here, work closely with these two colleges and bring up a package that would work and be sustainable. And then after we extend, but we are very much interested to know more about what's going on in other regions, such as yours.
Yes.
And also we want to engage more with Maori as well. So we have a very good connection in Northland, actually, with Patuarakeke there. And so we are also exploring opportunities to, to bring these sort of technologies, in particular DNA, eDNA. I think we need to inform young Maori kids, more about this, get them excited,
Yes.
and communicate. So these sort of things, yes.
Yeah, we have a few programs here. So I suppose it could be sending through your email to some of our contacts and getting them to get in contact. Would that be the best way? Or would you want the department to lead through that?
Let's start with email. Just reach out to me, and then yep, we can have another discussion maybe.
Okay, great. Thank you.
Thank you,
Natalia.
Natalia.
Yeah.
That's great, and great to have Doc involved as they are in both the north, top of the north and the top of the south partnership. So it's
Just
really been building over the last couple of years. But Ollie,
just to say, Natalia, and also acknowledging Doc, we have Monique Latz from Doc's office in Wellington. She's involved with us as well, and Doc have really kindly provided a lot of support for the program. We have all the visits to the Kermadec and Subantarctic Islands for the last few years were collated by Doc and provided to us to incorporate into this pathway model, so we can have the total, the complete spatial domain of New Zealand. So yeah, just wanted to acknowledge that, and we really enjoy interacting with you guys.
Oh, awesome. That's great to hear. Yes, Monique's lovely. We have some distributed staff in our office. So that's probably an advantage for myself as a, I guess what we call cold face ranger. It's good to be able to walk across the room and get information from our distributed marine staff, and very enthusiastic marine environment, of course, with the Marine Coastal Station there, very active iwi. So yeah, and it's great to go to hear a presentation, and it's still really valuable. So that was fantastic.
all right, thank you. And our time's up. But thank you both for all the work that you put into this. And I'm sure you're happy for people to look on your website and make direct contact. And, as I'm aware, you've got, this was just 2 sample bites. You guys are really busy. You've got lots and lots of things that people may want to have a look at. So thank you so much. And the recording will be up on our websites within the week.
Thank you, Peter.
Thank you, everybody.
Thank you, everyone.
Thank you.
Happy to.